Chapter 2, Atoms, Ions, and Molecules, Part 5, Nucleic Acids. Nucleic acids are the macromolecules that store genetic information in the nucleus and then transfer some of that information out to make proteins. There are two main kinds of nucleic acids. You have the DNA molecules and the RNA molecules. DNA is known as deoxyribonucleic acid because it uses the deoxyribose sugar, and RNA is known as the ribonucleic acid because it uses ribose sugar. And the key monomer for nucleic acids is the nucleotide. The nucleotides form long chains for either type of nucleic acids, and the nucleotides are connected to each other by phosphodiester bonds, covalent bonds between the phosphate group and the sugar. The nucleotide is made up of three main components. You have the phosphate group, you have the sugar, and you have the nitrogenous base. So again, phosphate group, sugar, and nitrogenous base. And there turns out to be 5 different nitrogenous bases. So they are cytosine, thymine, uracil, adenine, and guanine. You do not have to remember what they look like chemically, but you do need to know cytosine C is in DNA and RNA, adenine A is in DNA and RNA, guanine G is in DNA and RNA, thymine T is only in DNA, uracil U is only in RNA. So deoxyribose nucleic acid, found in the nucleus and in the mitochondria. It has the deoxyribose sugar and adenine, guanine, cytosine, and thymine. And the 2 strands of DNA, because DNA is made up of 2 long strands, long polymers, are connected to each other by hydrogen bonds between the bases, hydrogen bonds between the nitrogenous bases. And it's always connected by certain pairings. So thymine is always paired with adenine, guanine is always paired to cytosine. So if one strand has an A, then the other strand that base pairs with it will have a T. If it has a C, then the other one will have a G. So again, in DNA, A to T and C to G. Ribonucleic acid, RNA, is structurally very similar, except instead of thymine, it has uracils. Now, RNA is usually a long, single-stranded chain of nucleotides. However, those nucleotides can form complementary base pairs, either within the RNA molecule or with other molecules. But in this case, uracil now is going to pair with adenine. So replace the T-thymine with U-uracil. So uracil pairs with adenine, guanine pairs with cytosine. RNA is found in the nucleus, mitochondria, and in the cytoplasm of the cell. Another nucleic acid is adenosine triphosphate. This is basically an adenosine base attached to a ribose sugar attached to 3 phosphate groups. So triphosphate, 3 phosphate groups. Turns out that chemical bond between the second and third phosphate groups is a high energy bond. And when it's broken, it releases chemical energy. So the energy currency of the cell is ATP. So mitochondria makes lots and lots of ATP, and that is used for various chemical processes that need energy in our cells. There are a few other important nucleotides. We're not going to remember these names, but NAD and FAD, they show up very importantly in the aerobic cellular respiration. So in that process of turning glucose into lots and lots of ATP, they act as carrier molecules. They will carry energy around within the mitochondria that then eventually gets used to make even more ATP. So NAD plus and FAD. And here we can see DNA and RNA in the nucleus. RNA leaves the nucleus to go into cytoplasm, and they are very important for a lot of cellular processes. All right, complementary base pairing is something you're going to need to remember. So remember that T's bind with A's, C's to G's, and so on. U's to A's, A's to U's, C's to G's, and so on. And if U's are present in the molecule, then it must be RNA. If there are T's then it must be DNA. Proteins. Proteins are the most functionally important macromolecule in cells and in our bodies. They do a huge range of functions. They act as enzymes, which catalyze chemical reactions. So enzymes speed up chemical reactions so they occur fast enough to be relevant for our cells. Proteins can act in defense. Antibodies can bind to pathogens and help inactivate them or target them for destruction. Proteins can aid in transportation. For instance, proteins help move hydrophobic substances in the bloodstream. Proteins can produce structural support, forming actual protein structures, protein rods that can form shape. Proteins can cause movement, and proteins can perform regulating processes in the body, and they can be involved in storage. So the general structure of a protein is one or more monomers, the monomers in proteins being amino acids. So technically one amino acid is called an amino acid, not a protein. But proteins are made up of long strands of amino acids. Amino acids always have an amine group, a carboxylic group, and an extra R group off of the central carbon. And that R group has up to 20 possibilities. And that's how the different amino acids are different from each other. They're different by the R group. So when you get a long chain of amino acids, that means one amino acid's amine group is forming a chemical bond with another amino acid's carboxylic group. So that bond between a carboxylic group and an amine group is referred to as a peptide bond. Obviously, since we're attaching monomers together, this is a dehydration synthesis reaction. If you have 3 to 20 amino acids, it's called an oligopeptide. If you have 21 to 199 amino acids, it's called a polypeptide. And if you have 200 or more amino acids, it's referred to as a protein. Some proteins are made up of 2 or more polypeptides. Some proteins are just an incredibly long chain of amino acids. And this would be an example of a, looks like an oligopeptide. Again, the amino acids have different R groups. There are 20 possible R groups. Some of them are nonpolar, some are polar, some are charged, which technically is also polar, and some have other very specific structural arrangements. And all of these can interact with each other and other parts of the polypeptide to alter the shape, influence the function of the protein. You do not need to learn the names of all of these. You just need to know that there are 20 of them that do important things. So, primary structure of proteins is the linear sequence of amino acids. So just knowing the name of each amino acid along the chain is the primary structure. The secondary structure is how the different parts of the long chain of amino acids are interacting with each other, either forming alpha helixes that are helical in shape, a nice coil, or beta sheets, which are more pleated planes. These interactions are occurring via hydrogen bonding. So hydrogen bonding within the long amino acid chain leads to secondary structures, alpha helixes and beta sheets. Tertiary structure is a more three-dimensional structure of the protein. It can get extremely complex. The 2 big categories for it is globular proteins, which are compact in shape, and fibrous proteins, which tend to be long proteins. And again, this is interactions between various structures and various secondary structures in the protein, involving hydrogen bonding and other kinds of interactions between R groups. And this is the protein conformation, which is very important for protein function. So some of those intermolecular interactions include hydrophobic interactions between nonpolar R groups, hydrogen bonding, ionic bonds between charged R groups, and disulfide bonds between specifically the cysteine amino acids, which happen to have sulfur. You need sulfur for a disulfide bond. All right, quaternary structure. Quaternary structure occurs in some proteins, not all proteins. Quaternary structure is when you have two or more polypeptide chains necessary to produce a functional protein. So here's a globular protein with 4 polypeptides, a fibrous protein with 3. So again, quaternary structure, 2 or more separate polypeptides forming the functional protein, only present in some proteins, not all of them. All of them have primary, secondary, tertiary, some have quaternary. Enzymes, as we know, are very important because they are catalyzing chemical reactions. They have an active site where a specific substrate will bind, and then it will allow for the speed up of a chemical reaction. So for instance, down here, we have an enzyme with 2 substrates coming together in the active site. I'm sorry, I said that wrong. You'll have an enzyme with 1 substrate binding to the active site. Chemical reaction happens, and that substrate gets broken into 2 products. What happens specifically is enzymes lower the activation energy. So if you have your reactants, your substrates, they have to go and overcome a major mountain, a major large amount of activation energy in order to lead to the products forming. This, say, may take thousands of years to occur, not very useful for our cells. So through the help of enzymes, it lowers the activation energy so that now the reactants become the products in, say, milliseconds. And that is relevant for life. So enzymes catalyze the chemical reaction. Turns out that also means enzymes do not get used up by the chemical reaction. You have them present at both the reactant side and the product side, and then they can get used again and again and again to speed up chemical reactions. And again, the enzymes are very specific. They only accept certain substrate or substrates. This is determined by the shape of the active site and the shape of the substrates. So often referred to as a lock and key mechanism. Only a key shaped properly will fit into a lock to unlock it. So active site binds to the enzyme, substrates bind to the active site, giving us an enzyme substrate complex, which then speeds up the chemical reaction, leading to the product or products, and the unaltered enzyme ready to get used again. Again, enzymes are highly specific, so they only do one thing. Denaturation. Denaturation means there's a conformational change in the protein. This is a permanent change to the shape of the protein and is irreversible and destroys the active site. No active site, protein no longer functions. This can be done by heat, by heating things up, by changing pH significantly, by radiation, by other chemicals, basically damaging intramolecular interactions and permanently inactivating the protein. And proteins, as we know, are very complicated, very important, found in cells, make up the cells, are used in the bloodstream. They're just everywhere. They're very critically important. And that is it for this chapter.